Hello and welcome to my channel Omni Bio Entrance. If you are new to the channel, please do like, share and subscribe. I will be doing University of Hyderabad PhD entrance examination question paper for the subject biochemistry. I will be doing only the biology part. This was conducted in the year 2022. Coming to the first question. Aspartate ammonia lyase would catalyze which one of the following reaction? So aspartate ammonia lyase, it catalyzes the reversible deamination of L-aspartate yielding fumarate and ammonia. So the answer here is deamination. Answer is option B. Then next one. From the Lin-Weaver-Burke plot of inhibition studies of an enzyme, it was concluded that it is an uncompetitive inhibition. Which one of the following observation would have directed to the conclusion? So there will be decrease in both Vmax as well as Km, and it is an uncompetitive inhibitor, so it binds to the enzyme substrate complex. So the answer here is option A. The following statements are related to lysosomes. Choose the combination that has all the correct answers. So the lysosomal lumen pH is approximately 4.5 to 5. That's correct. Centrifugation at 1 lakh into G can be used to isolate a lysosomal fraction from the liver cell extract. It is not 1 lakh, it is 15,000 G for 15 minutes. So option 2 is not correct. Phosphatase, esterases, nucleases, sulfates, phosphatases, lipases and glycosidases are the components of lysosomes. That's correct. Lysosomes are involved in autophagy. That is also correct. So 1, 3 and 4 are correct and the answer is option D. Then next one. You would like to determine the contamination of bacterial or mitochondrial ribosome in cell-free translation systems you prepared from an eukaryote source. To achieve this, the best way would be to determine protein synthesis in the system in the presence of chloramphenicol antibiotic. Answer is option D. Which one of the following organelle is enclosed by a single membrane? It is your endoplasmic reticulum out of the given options. So answer is option D. What is peptide anticodon in protein synthesis? So a peptide anticodon is a sequence of specific amino acid present in release factor 1 involved in the termination of protein synthesis. Answer is option C. Then which one of these proteins serve as guanine nucleotide exchange factor? So bacterial elongation factor EFTS. Option C. All these are just direct questions. That's why I am not going much into explanation. Then next one. A globular protein in a cell in a certain experimental condition aggregates to form either a tetrahedral tetramer or a linear tetramer. In the mixture, if the mixture is chromatographed on size exclusion chromatography, which will elute first? So the linear form will elute first. Why? Because the linear molecule cannot enter the beads. So the answer here is option B. The next one. When X-rays of wavelength 1.5 angstrom are scattered by a protein crystal, which one of the following conditions is required to observe a peak of diffraction intensity? So the answer here is the path length difference between the two scattered lights is equal to 1.5 angstrom. Answer is option B. The next one. The applied centrifugal field at a point 5 centimeter. In a cell from the center of rotation of centrifuge whose angular velocity is 4000 radians per second is G is equal to omega square R. So substitute the values. Omega square is your angular velocity, R is the radius. So 4000 square into 5, that will give you 8 into 10 raised to 7 radians square centimeter second raised to -1. So the answer here is option C. Peptide bond geometry can be estimated by X-ray diffraction, XRD. Answer is option C. MicroRNAs are important gene regulators, but the miRNAs are also regulated in turn by other RNAs. Which of the following classes of RNA are known to regulate miRNAs? So they are long non-coding RNAs which regulate miRNAs. Then next one. Identify the correct statement about the fate of carbon skeleton of amino acids. So glucogenic amino acids can give rise to glucose. That's correct. Ketogenic amino acids can give rise to acetyl-CoA. That's also correct. Ketogenic amino acids can be converted to mevalonate. That is not correct. Some amino acids are both glucogenic and ketogenic. That is also correct. So the answer here is option D, 1, 2 and 4. The next one. Using hepatocyte as a model system, Sutherland examined the effect of epinephrine on glycogen breakdown and measured various biochemical activities of the liver extracts A to D. The correct order of sequence of events representing epinephrine-mediated glycogen breakdown in the liver cell is glucose synthesis, CAMP production, protein phosphorylation, adenylate cyclase activation. So first will be adenylate cyclase activation, then comes CAMP production. Once the adenyl cyclase is activated, CAMP production starts. Then comes protein phosphorylation and then glucose synthesis. So the answer here is DBCA. DBCA comes in option C. Answer is option C. The next one. Match the following mammalian histone variants with their predominant roles. So H2AZ, it is active transcription promoters. Then CENPH3, it is centromeric histones. 2E1D. Macro H2A, it is X chromosome inactivation. 3A. H2AX, it is DNA damage signaling. Answer is option B. And H34B. H33 is 5C, male gametogenesis. So the answer here is option A. 1D, 2E, 3A, 4B and 5C. The next one. In order to determine the genes involved in non-homologous end-joining mediated DNA double-strand break repair, you perform a plasmid rejoining assay where you transform each mutant cell either with a cut plasmid or an uncut circular plasmid and then score for the efficiency of end-joining in that particular mutant background. This assay works on the principle that once the cut plasmid is rejoined within the cell to produce circular plasmid, then only transformant is obtained. From the accompanying graph, infer the genes that are likely to be involved in NHEJ. Mutant genotype is written as small letter gene P and wild type genotype is written as capital letters gene P, etc. So if you see the graph, you can see that maximum transformation, that is number of transformants obtained, is more in the case of gene P and gene Q. Is more for them. So the answer here will be gene P and gene Q. Answer is option B. The next one. Following are the phenotypes of two temperature sensitive mutants of two essential genes ESS2 and ESS5. Plus indicates ability to grow at that temperature. What would be the phenotype of an ESS2 ESS5 double mutant? The double mutant will be inviable at all these three temperatures. It will not grow. Answer is option D. The bacterial cell catabolite repression allows the cell to save energy by. So it allows the cell to save energy by regulating expression of genes required for utilization of less efficient metabolites. Answer is option C. Coming to the next question. The following Ramachandran plot most corresponds to. So if you see the Ramachandran plot over here, you have got the maximum over the beta sheet structure. Beta sheets are more prominent here, and beta sheets are prominent in beta barrels. So it corresponds to beta barrel protein. So the answer here is option B. Then many biochemical reactions are thermodynamically unfavorable. However, they occur in cells through coupling with other reactions, especially the reaction ATP giving ADP plus PI. Based on this information, which of the following can be true? Delta G for ATP hydrolysis is negative. Answer is option B. Then next one. The table below has columns indicating the immune mediators and types of hypersensitivity. So IgG, IgM mediated complement activation and antibody dependent cell cytotoxicity. It is type 2. Type 2. Then IgE mediated mast cell and basophil degranulation. It is type 1. Sensitized T cells, that is type 4. And Ig bound soluble antigens forming immune complexes, that is type 3. So the answer here is option C, A2, B1, C4, D3. Then coming to part B. A physician prescribed statin for his patient suffering from myocardial infarction. Which of the following would be the specific activity of statin? So statin, they work by competitively blocking the active site of the enzyme HMG-CoA reductase, and what will happen once HMG-CoA reductase is blocked? It will block the conversion of HMG-CoA to mevalonic acid. So a physician prescribed statin to his patient suffering from myocardial infarction. Which of the following would be the specific activity of statin? So statin inhibit HMG-CoA reductase. Answer is option D. Then identify the correct statement about the reaction catalyzed by ribonucleotide reductase. So RNR, it is an enzyme responsible for the conversion of ribonucleotides to 2'-deoxyribonucleotides, or RNR converts NDP to DNDP. Answer is option B. Then micronutrient essential for synthesis of purine is folic acid. Answer is option A. Then which one of the following biochemical events ensures the translocation of electrons produced into mitochondrion during glycolysis? So it is the malate aspartate shuttle, and it is responsible for translocating electrons produced during glycolysis across the semi-permeable inner membrane of mitochondria, and the electrons take part in oxidative phosphorylation in eukaryotes. So the answer here is option C. Then which one of the following post-translational modifications of histone proteins are recognized by chromodomain containing proteins? So it is methylation. Answer is option A. Chromodomain containing proteins are responsible for gene regulation. Then next one. Which of the following events is not involved in eukaryotic mRNA splicing? Formation of spliceosome complex, that is involved. Formation of 2' 5' phosphodiester bond, yes. Formation of lariat intermediates, yes. Coupling of phosphodiester formation to ATP hydrolysis, that is not involved. So the answer is option D. Then next one. Increasing the concentration of which of the following would effectively antagonize the inhibition of protein synthesis by puromycin? So puromycin, it causes premature chain termination during translation, and the part of this puromycin, it resembles aminoacylated tRNA. It enters into the A site and transfers to the growing chain, causing premature chain termination. So they have asked which would effectively antagonize the inhibition of protein synthesis by puromycin. I told you already, puromycin, part of it resembles aminoacylated tRNA. So aminoacyl tRNAs would be the answer here. The next one. Anaphase promoting complex has which one of the following enzymatic activity? So it is ubiquitin ligase. Answer is option C. Then next one. The CpG islands, the site of DNA methylation in mammalian genomes are. So they are present less often than expected by random chance. Answer is option C. Then next one. Which one of the following organelles contain DNA? It's a direct question. The nucleus, mitochondria and chloroplast. Answer is option D. Then next one. Which of the following technique is best suited to estimate molecular weight of a given protein? So naturally it is SDS polyacrylamide gel electrophoresis. Isoelectric focusing is for determining the PI. Bradford is for qualitative assay. Then EMSA is for determining DNA protein interaction. So answer here is SDS polyacrylamide gel electrophoresis. Then next one. Which of the following mutagens causes frame shift mutation predominantly? So it is acridine orange. It is an intercalator. So it causes frame shift mutation, either insertion or deletions. Then next one. Non-coding DNA in eukaryotic cell does not include. So it does not include simple sequence repeats. Arachidonic acid is an omega-6 fatty acid. Answer is option B. Which of the following is a C3 plant? So out of these, rice is a C3 plant. All the others are C4 plants. Then next one. Which of the following is a tandem enzyme? So phosphofructokinase 2 is a tandem enzyme, and tandem enzymes are also called as bifunctional enzymes. And what are tandem enzymes? So they are a pair of opposing enzymatic activities that are resident in the same polypeptide chain. So phosphofructokinase 2 is a bifunctional enzyme because fructose 6 biphosphate can be converted to fructose 6 phosphate by phosphofructokinase 2, and fructose 2,6 biphosphatase convert fructose 6 biphosphate can be converted to fructose 6 phosphate by fructose 2,6 biphosphatase, and fructose 6 phosphate can be converted to fructose 6 biphosphate by phosphofructokinase 2. So these two activities of the enzyme are present in the same polypeptide chain, even though their action are opposing to each other. So phosphofructokinase 2 is a tandem enzyme or a bifunctional enzyme. Then next one. Which of the following two enzymes are glyoxylate cycle specific enzymes? So the answer here is option B, that is isocitrate lyase and malate synthase. Then next one. The core oligosaccharide added to asparagine residues in N-glycan synthesis contain. So it contains 2 N-acetyl glucosamine, 9 mannose and 3 glucose residues. To produce one urea molecule via urea cycle, dash is required. So how many ATPs are required? So the first step of urea cycle, it utilizes 2 ATP which is converted to 2 ADP plus 2 PPI for the formation of carbamoyl phosphate, and the third step that utilizes 1 ATP which is converted into AMP plus PPI for the formation of araginosuccinate. So totally 3 ATP are required. Answer is option B. Then next one. Glutamine synthetase is regulated reversibly by. So it is regulated reversibly by adenylation and deadenylation. Answer is option A. Then next one. The gene for eye color in dog has 2 alleles, capital B and small b. Capital B is dominant and codes for black eye. Small b is recessive and codes for grey eyes. The possible genotype and phenotypes in this population of dogs are. So there is only one particular character involved here, that is color. So color, that is either black or grey. There are no intermediate colors. So the phenotype will be black and gray, and the genotypes can be capital B, capital B, capital B, small b, and small b, small b. So the answer here is option A. The next one. In pea plants, round seed are dominant over wrinkled seeds. If a plant that produces round seeds came from a parent that produced wrinkled seeds, what is the genotype of this plant? So naturally it is heterozygous because it produces round seeds coming from a parent with wrinkled seeds. So naturally it is a heterozygote. So the genotype will be small r, capital R, small r. The next one. In a family, the mother has a defect in meiosis leading to high level of non-disjunction. Therefore, the eggs gametes either contained both X chromosomes or no X chromosomes. In the surviving children from this mating, the number of bar bodies produced will be either 0 bar bodies or 1 or 2 bar bodies will be produced. Answer is option D. Coming to the next question. Wild type yeast can be stored in low water or low solute solution for days without loss of viability. A yeast mutant defective in cell wall biosynthesis. Defective in cell wall biosynthesis was isolated. When this mutant is stored in water, what would you expect? So wild type yeast, it is having a cell wall. So it will remain in water and low solute containing solution for days without loss of viability because osmosis will not take place. But yeast mutant defective in cell wall biosynthesis, osmosis takes place and cell will swell and burst. So the answer here is option B. Then next one. A protein is synthesized on cytosolic ribosomes. In a pulse chase experiment to trace the trafficking of this protein within the cell, one is likely to find this protein in the nucleus. Next one. Which one of the following statements about ion exchange chromatography is not true? Binding of protein to a column is done in low ionic strength and eluted at higher ionic strength. That is true. They have asked for not true. To bind a protein to a cation exchanger, binding is carried out at pH lower than the pI of the protein. That is also correct. Protein bind to exchange resins via non-covalent ionic interactions. That is also correct. In ion exchange chromatography, the active group on the column are negatively charged. They are not negatively charged, they are positively charged. So the incorrect statement here is option D. The next one. The following equation is used in blast scoring statistics. S dash is equal to lambda S ln k by ln 2. This equation denotes bit score. Answer is option C. Next one. Which of the following microscopes can be used for studying the ultrastructure of mitochondrial inner membrane and resolving structures that may be just approximately 10 nanometers to 50 nanometers apart? So TEM can be used. That is transmission electron microscopy. The next one. The GTP bound form of RAC1 GTPase promotes actin filament formation. GAPs are proteins that increase GTPase activity of small GTPases. Overexpression of GAP specific for RAC1 will likely. It will decrease the ratio of filamentous actin to monomeric actin. Answer is option D. The next one. Which of the following reagents are mandatory requirement for you if you are required to measure the quantity of antibodies present in patient serum against a toxin from a pathogen? Enzyme conjugated antibody against human IgG, that is required. Purified toxin of the pathogen, that is also required. Polyclonal human IgG against the pathogen toxin, no. So answer here is option C, that is 1 and 2 are required. The next one. Under which of the given conditions might an antigen presenting cell bind an antigen for presenting through MHC class 2 molecule? So when APC, that is antigen presenting cell, is treated with paraformaldehyde about 2 hours post antigen exposure. Yes. APC treated with paraformaldehyde and then incubated with native antigen. No. It should be post-antigen exposure. Then next one, when APC is treated with paraformaldehyde and then incubated with antigen, partially digested with the enzyme. That is also correct. So the answer here is option D, that is both A and C are correct. Then next one. Which of the following statements are false about gram staining? Gram-positive bacteria retain the crystal violet stain because they have thick multi-layer sheet of peptidoglycan. Yes. Gram-negative bacteria do not retain crystal violet stain because they have thick layer of teichoic acid. Gram-negative bacteria doesn't have teichoic acid. So that statement is false about gram staining. So your answer is option B. The next one. Which of the following viruses carry reverse transcriptase enzyme in their capsid? They are retroviruses. Answer is option D. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.